Everyone, hi everyone, welcome to my channel Pure Biology, and today I come with important topic, that is the nitric oxide. How this nitric oxide acts as the paracrine signaling molecule. So paracrine signaling molecules are those molecules which acts on the target cells that are closely related with the molecule's secretion. Okay, or closely just located, you can say located, closely located with the molecule secretion. So these are called the paracrine signaling molecules. So NO or the nitric oxide, this acts as paracrine signaling molecule. Okay, so before going to this topic, I want to say that many topics related to cell signaling we already I just uploaded in my previous videos, so you just check these all. Okay, so here the NO is what? This is the paracrine signaling molecule, and this is able to diffuse directly across the plasma membrane. PM is the short form of the plasma membrane of its target cells. So what is saying that these secretes and diffuse directly across the target cells, which are very closely related with the molecule's secretion area. Okay, so here this is synthesized from where? From the amino acid arginine. Okay, this is synthesized from arginine. This is one kind of amino acid, and here the enzyme is very important, that is the nitric oxide synthase. This nitric oxide synthase actually, this amino acid arginine is the main source from which this nitric oxide is secreted, and here the enzyme is the nitric oxide synthase enzyme. Okay, nitric oxide synthase enzyme. And once this synthesized, that diffuses out of the cell and can act locally to affect the nearby cells. What I told, that it act on the neighboring cells or the nearby cells. Okay, just closely associated or closely located cells. These are the target cells of these paracrine signaling molecules. It cannot affect on the cells that are situated far away from the area of the secretion of the molecule. Okay, so here this is actually where after synthesis, what happening? After just synthesis, this diffuses out of the cell and can act locally. Okay, can act locally. Now example is that in mammals, NO action is to relax the smooth muscles, and this causes the dilation of the blood vessels. That means it helps in the relaxation of the smooth muscles and as well as the dilations of the blood vessels. Okay, so how the actual process is occurring? Okay, so first, in some cases the autonomic nerves in the blood vessel wall that release the acetylcholine. This is one kind of neurotransmitter. This acetylcholine acts on the endothelial cells that line the interior of the blood vessels. Okay, so what is saying that first acetylcholine is secreted from where? From the blood vessels wall, and this acts on what? This acts on the endothelial cells which are lined up with the interior of the blood vessels. Interior portion of the blood vessels, obviously. So to stimulate the NO synthesis. So this acetylcholine, it indirectly helps in the synthesis of the NO. Okay, or you can say it stimulates the NO synthesis. First, acetylcholine is released and this acts on the endothelial cells, and after that NO synthesis is occurring or continuing. Okay, and this NO diffuses the neighboring smooth muscle cells in the walls of the blood vessels. What is saying that NO where diffuses? This diffuses the neighboring smooth muscle cells in the wall of the blood vessels. So from the blood vessels which are associated with the endothelial cells, these blood vessels related smooth muscles or the smooth muscles attached with the blood vessels wall, this area is very important for the diffusion of the NO. Okay, and here it interacts with the iron. Okay, this NO after secretion, this interact with the iron. This iron, which iron these are? These iron are that bound to the active site of the enzyme guanylyl cyclase. Okay, not all iron, only those iron bound to the guanylyl cyclase. With that iron, the NO interacts. Clear? And this increase the enzymatic activity. Okay, this increase the enzymatic activity, and resulting is what? And synthesis of the second messenger, that is the cyclic GMP. Okay, so when this occurring, that means the interaction of the guanylyl cyclase and the AO is completed, then the synthesis of the second messenger cyclic GMP is occurring. Okay, or GMP secretion is seen there at that time. So which induces the muscle cell relaxation. So that means after the secretion or the synthesis of this cyclic GMP, the muscle cell relaxation and the blood vessel dilation is seen. Okay, that means after this secretion or synthesis, the cell relaxation, muscle cell relaxation and the blood vessel dilation is possible. Okay, both are possible. So here this guanylyl cyclase acts as in two ways. One, as signaling protein and also receptor. Clear? Intracellular receptors as well as intracellular signaling protein. So this is happening actually due to the NO synthesis. Okay, this is happening actually due to the NO synthesis. Okay, so how NO helps in the blood vessel dilation and the relaxation of the muscle cell, we just learned here. Okay, and sometimes NO is also produced by the activated macrophages and the neutrophils for actually killing the microorganisms. Okay, so NO also sometimes help these neutrophils and macrophages to kill the microorganisms. So this is another important topic, obviously. That means how this nitric oxide acts as paracrine signaling molecule. It is obviously, this is another topic not related with the receptors and the pathways of the cell signaling process. This is you can say this is one kind of example how the signaling process is done. Okay, so in that way there are many examples or many situations occurring due to the signaling process, cell signaling process. So we will learn about that topic in my next videos. Clear? So please you just share and subscribe my channel to give your support, and I will try to give my best for you. Okay, so thank you.